So, we're looking at Urey and Miller had to assume, contrary to the opinions of geologists, that the early Earth had no oxygen in its atmosphere. This is because amino acids are destroyed by oxygen. But absence of oxygen implies an absence of ozone, another form of oxygen. Ozone in our atmosphere protects us from high-energy ultraviolet rays from the sun. Nucleic acids are rapidly decomposed by ultraviolet light. A further difficulty for those who postulate abiogenic synthesis is that the molecules formed are destroyed by the very conditions, such as heat, UV light, and electricity, that make them. Proteins and polynucleotides are thermodynamically unstable. They are also unstable with regard to hydrolysis and reactions with other simple reagents. Moreover, the longer the experiment is allowed to continue, the more decomposition products are made. Many processes are reversible, but in the equilibrium state, simpler, startling materials predominate over more complex products. Time does not help the forward reaction. The tarry messes from these experiments are in stark contrast to the neat metabolic pathways of living cells with their clean, high yields of precisely fashioned products. In their experiments to simulate, sorry about that interruption. The tarry messes from these experiments are in stark contrast, as I said, to the neat metabolic pathways of living cells with their clean, high yields of precisely fashioned products. In their experiments to simulate abiogenic development, investigators begin each stage with pure compounds in high concentrations. This can hardly reflect natural conditions on a prebiotic Earth. In the living cell, the DNA codes for proteins, and the DNA is itself constructed using proteins. It is a chicken and egg situation. It has been suggested that RNA processes some of the enzymatic properties of proteins while having the information carrying ability of DNA. Could positing a first protocell that relied on RNA for both functions solve the problem? No primeval soup experiment has ever produced anything resembling RNA. RNA does not replicate itself, a prime necessity for a living cell. RNA's enzymatic properties are not sufficiently versatile for even the simplest imaginable protocell. The problem of the origin of information on the information carrying RNA remains unsolved. A protocell based solely on proteins is equally impossible, since proteins lack the ability to reproduce themselves. Sound like an intelligent designer here? Each component of a living cell is breathtakingly complex, yet in isolation it cannot survive nor replicate itself. All the parts of the cell are necessary to its functioning and replication. Nothing works until everything works. This has been called irreducible complexity. Even small parts of the components of cells can be unimaginably complex. An example of this is the enzyme adenosine triphosphate synthase, found in all living cells, including animals, plants, fungi, and bacteria. The elucidation of the structure of ATP synthase won a 1997 Nobel Prize. Every cell contains hundreds of these miniature motors embedded in the surfaces of the mitochondria. Each is 200,000 times smaller than a pinhead. The motor forges a bond between ADP and phosphate to form ATP. The ATP couples with other processes in the cell requiring energy to reform ADP and phosphate. So energy is directed to contract muscles, beat the heart, and drive thought processes in the brain, while the products are recycled. At the center of ATP synthase is a tiny wheel that turns at about 100 revolutions per second and turns out 3 ATP molecules per rotation. Just to keep us thinking and walking, animals, humans, must recycle their own body weight of ATP each day. Each enzyme is composed of 31 separate proteins that in turn are made of thousands of precisely arranged amino acids. Take away any one of the 31 proteins and the motor is useless. It could not have evolved. And consider this: the genetic information and RNA plus proteins needed to construct the ATP synthase are in total even more irreducibly complex than the ATP synthase itself. A car-making factory is more complex than a car, to give you an idea. The concept of abiogenesis is vital to the atheistic evolutionists. It follows in their thinking that if life can arise spontaneously under the right conditions, then there will be perhaps millions of planets in the universe where life already exists. In some of these places, intelligent life may have evolved. These ideas have spawned a large body of literature, films, and video games involving imaginary extraterrestrial life. Billions of dollars have been spent by government-sponsored searches for messages from out there, e.g. Project SETI. The irony is that evolutionists would recognize that a non-random signal from space that carried information with meaning and purpose must have come from an intelligent extraterrestrial. Yet they consider nucleic acids in the living cell a non-random sequence of nucleotides carrying far more information with precise meaning and exquisite purpose, and say it must have arrived by chance. So, there you go.